Hi, my name is Alan, and today I'll be presenting on our method to use a matrix prior for data transfer between single-cell data types in latent Dirichlet allocation. We'll start with some background about what latent Dirichlet allocation is and what our motivation for this project was. Latent Dirichlet allocation, or LDA, was an algorithm historically used to model text corpora, made up of many different documents. Here we have three. Cats are really furry and purr often. Dogs love walks and to fetch and catch. And cat-dog is a conjoined twin. The goal of LDA then would be to tell us that first, document 1 is primarily a cat document. Document 2 is primarily a dog document. And document 3 is a cat-dog document, a mixture of both cat and dog. How latent Dirichlet allocation accomplishes this is by assigning every word to one of two topics in this case, although generally we can have as many topics as we'd like. Here we might expect cats, furry, and purr to be cat words. On the other hand, dog, fetch, and walks are dog words. That is, unless you have a particularly adventurous cat. So although historically LDA was used on text data, LDA has actually recently been adopted in the single-cell data modeling technology as well. In these cases, we would use each cell as a document and either a peak from SCATAC-seq or a gene from SCARNA-seq as a word. Our contribution is that we created a prior to transfer information between datasets using LDA, and we were motivated by the transfer information from a reference SCATAC-seq dataset to a smaller target SCATAC-seq dataset, which would allow us to improve resolution of cell types in the target SCATAC-seq data using this auxiliary data. So how does the LDA algorithm work? Well, one approach to it is using the collapsed Gibbs sampling approach. Here, we assign for each cell in a gene, we would assign it a topic, and the probability of it being assigned a topic K is proportional to first, how many times the cell C has this topic K, and then also how often a gene G shows up in topic K. In addition to this, we have prior information from our Dirichlet in the form of alpha and beta. Our contribution is that currently, in the modern form of LDA, beta is a vector. We propose to change it to a matrix that depends on the topic as well to carry more information. Here we see that the form is quite similar, except now beta is actually dependent on K, the topic. The rest is quite similar. The probability of a gene being assigned a topic K is proportional again to the number of times that this topic appears in the cell, how often this gene shows up, and again we have these priors that are being added with the new addition that beta is now a matrix. We use this method to apply to some C. elegans data with SCATAC-seq. We started with the full data set. We simulated having a target data set and a reference data set by dividing this data into two. Then, we started by analyzing the reference data set using a uniform prior to achieve a reference topic gene matrix and a reference cell topic matrix. When we now analyze our target dataset, we can now use the topic gene matrix from our reference data as a prior for the target data. This results in having the green target topic gene matrix and cell topic matrix. In addition to that, we can compare this to the full data set when we've analyzed all of the data together, which we consider to be kind of a gold standard, which is more computationally intensive and a little less flexible. When we compare these, we simply stretch them out and plot them against each other to see if we're capturing similar structure in the target as when we did the joint analysis. In this figure, we can see that when we use the uniform prior on our target data on the very left-hand side, we see that many of the points in the cell topic matrix are floating around and not quite aligned with what the cell topic matrix is when we did the joint analysis on the x-axis. Similarly, for the topic gene matrix, we can see kind of a scattered pattern on the very left-hand side where we've used a uniform prior. On the other hand, as we increase the weight of our prior to the right, we see that we have better matching between our target dataset and the true joint dataset. Finally, we look at UMaps of these data, both with a uniform prior and a reference prior, and we see that there is some additional substructure that comes out using our reference prior. In particular, on the bottom panel, we see that some of the neurons are better separated. So in summary, we used LDA to modify this algorithm to include a matrix prior that includes information about different topics, and we were able to more accurately capture cell topic distributions and uncover fine-grained cell type information. Thanks for listening.